So I'd like to thank the organisers for giving me this opportunity to present my work today. So it was Chuck Peru and colleagues who showed in the early 2000s that tumour phenotypic diversity could be correlated with patterns in global gene expression. And they identified 5 intrinsic gene signatures which could not only be used to predict molecular subgroups, but it also could predict outcome, with the luminal subgroup doing the best and the triple negative the poorest. But despite this and many subsequent studies, over 685,000 women are still dying from breast cancer each year. And this demands that we need to better understand what drives poor outcome even within these molecular subgroups. So it is becoming ever more obvious that the immune system plays a key role in cancer. Not only can it eliminate cancers, but it can also shape cancers and edit them through putting on selective pressure, which leads to immune evasion. And this immune evasion can happen at any point throughout the cancer immunity cycle. This in turn leads to 3 distinct immune phenotypes that are observed in cancer. As shown here on the right-hand side, we have these immune deserts, where really the immune system has no real knowledge of a cancer being present, and so no reaction has been mounted. In the middle, we have these cold or excluded tumours, where the immune system has recognised the tumour, has tried to mount a response, but it's kept excluded from the tumour microenvironment, or the TME, probably by secreted factors. Here on the left-hand side, we also have these inflamed or hot tumours, where again, the immune system has recognised the tumour and mounted a response, and we can find tumour cells or immune cells right within the tumour. Quite often these immune cells, however, are kept in check by the presence of immune checkpoint proteins. It is these hot tumours that are associated with the best outcome, and this can be shown looking at or correlating the amount of TILs or tumour infiltrating lymphocytes with it in cancer. And high TILs are associated with improved outcome in breast cancer, especially within triple negative breast cancer, where you can see patients with the highest amount of TILs do the best. So therefore, we really need to better understand what drives these TILs into the tumour to make a hot or inflamed tumour, and how can we best capture this tumour microenvironment, taking into account both good and bad immune cells and immune checkpoints. So recently, the STING pathway has emerged as being very important in cancer. It is actually part of the ancient antiviral pathway, which is used to detect cytoplasmic DNA. This triggers the cGAS pathway, or protein which produces the secondary messenger cGAMP, which goes on to activate STING, and this leads to an interferon response, which activates the immune system and brings immune cells right into close proximity with the cell so it can be eliminated. Cytoplasmic DNA can also be found in cancer because of maybe the high rate of mutations and proliferation found within cancer cells. And this can also trigger a STING response, also leading to an interferon response and attraction of immune cells into the tumour. So what do we see of STING in breast cancer? So using immunohistochemistry to visualize STING expression, we saw 4 distinct expression patterns. We either had tumours where we have both high and low, or high expression of STING both within the tumour and stroma, high expression in either the tumour or the stroma only, and then also some tumours with low expression altogether. In order to best capture this and quantify it, we use the open source software called QPath to be able to best digitally quantify this in a robust and reliable manner. When we looked, however, at the patterns of expression of STING within the tumour microenvironment, or within the tumour, we saw occasionally these tumours with this kind of distinct pattern of STING expression within or around the nucleus in this perinuclear space. And it's very similar to what we see in cells when we add cGAMP. And in fact, when we look at how STING signalling actually occurs, what happens is that when STING is activated, it translocates from the endoplasmic reticulum to this ergic region, and this leads to this distinct perinuclear expression pattern. So therefore, the localization of STING in this perinuclear space may actually reflect active STING. And so we wanted to then capture this perinuclear localization and thus potentially STING activity within our tumour samples. So we developed a bespoke digital pathology pipeline, again using QPath, to not only capture, I said, tumour and stromal STING expression, but also to capture this perinuclear expression. We then applied these pipelines to a cohort of breast cancer patients that had all been treated with standard of care anthracycline-based chemotherapy. We had a matched tissue TMA resource, as well as transcriptomics, as well as clinical and pathological data with at least 5 years follow-up. So when we looked at this, we could see that most patients, or most cases, had high expression of stromal STING, while there was a kind of a broader range of tumour expression of STING, and then some expression of perinuclear STING. When we looked at the correlation between these, we could see in general good correlation between high perinuclear STING and high stromal and tumour STING. This is a normalized heat map here, where red shows high expression and blue shows low expression. Looking then instead at what this might mean, what we saw when we correlated with outcome was that while all forms of STING expression were correlated with improved outcome, only this high perinuclear STING was significantly associated with outcome, where patients with low perinuclear STING were almost 2.5 times more likely to relapse compared to those patients with high perinuclear STING. So looking at this more closely within clinically relevant subgroups, what we saw was that this pattern of perinuclear STING being associated with outcome was only evident in ER positive breast cancer and not in the ER negative breast cancer, despite a very similar pattern of expression. So given that this expression was associated, or given that we could see that perinuclear STING was associated with outcome, and given the link of STING with immune signaling, we wanted to look in more detail at whether there was any correlation with the immune contexture. And to do this, we carried out immunohistochemistry on the same samples and looked for markers of macrophages, T cells, B cells, NK cells, as well as a number of immune checkpoints. And what we could see was that high perinuclear STING was positively associated with T cell markers and NK markers, indicating that it was associated with an inflamed or hot tumour microenvironment. But also we saw a high expression of immune checkpoints, saying that this probably was keeping this inflamed tumour microenvironment in check. As we had matched transcriptomics, we could further investigate the TME by deconvoluting this to give us, using a number of gene signatures. And what again we could see was that high perinuclear STING was associated with this inflamed tumour microenvironment. What was interesting was that these signatures allowed us to also see that low perinuclear sting or patients with low sting activity and poor outcome were associated with this higher expression of an M2 bad anti-tumor or pro-tumorigenic tumor microenvironment. So what we wanted to do then was to kind of look a little bit more to try and understand what might be driving this, and whether we could also see this in other settings. So in order to do this, we wanted to develop a gene signature that could capture our high and low perinuclear sting cases, so that we could look in additional data sets that we didn't have tissue availability for. And we developed a signature focusing on the genes that were overexpressed in low sting activity, as those genes associated with high activity were mainly immune genes and so could be activated by other pathways. So using this signature, we could see that, again, high perinuclear or high sting activity was associated with good outcome in ER-positive disease. And in the same match samples, you could see it was very similar to what we saw by using the immunohistochemistry. We then went on to apply this signature to additional samples within our in-house cohort that we didn't have IHC for, and once again could see the same pattern with high perinuclear sting defined by our gene expression signature associated with good outcome, but only in ER-positive disease. Given that our cases, our in-house cases were all treated with anthracycline-based chemotherapy, and ER-positive breast cancer cases can be treated with either endocrine treatment only or endocrine treatment plus chemo, we wanted to look in additional data sets to not only validate our findings, but also to understand whether the treatment whether this finding was dependent on treatment. So we used the Metabrick cohort available through CBioPortal, and looking at all ER-positive breast cancer could again show that high perinuclear sting as defined by our gene expression profile was associated with good outcome. We then broke down this patient cohort into patients treated with endocrine treatment only or endocrine plus chemo, and again could see, regardless of treatment, high sting activity was associated with good outcome. This was also seen in another two cohorts, the TCGA cohort and the Wang dataset, which was treated with endocrine treatment only. So we wanted to further explore kind of the molecular characteristics associated with this difference in outcome. So in order to do this, we carried out gene set enrichment analysis. And as expected, in patients with high sting activity and good outcome, we saw lots of enrichment for immune pathways. However, in these cases with low sting activity and poor outcome, we instead saw enrichment for cell cycle genes and also MYC pathways. This was backed up when we looked at the genomic data, either looking at copy number amplifications or indeed mutations, where we saw that low perinuclear signaling and therefore poor outcome was associated with MYC and cyclin D1 amplification, as well as increased mutant P53. This was especially interesting to us, given that there have been a number of publications linking either MYC or mutant P53 to suppression of immune signaling through sting. And this is summarized here in this diagram, where MYC can suppress, directly transcriptionally repress expression of SING or interferon signaling genes, as well as inhibiting JAK-STAT signaling. Mutant P53 has also been shown to directly interact with TBK1 and therefore inhibit STING signaling. So to summarize, we have developed a bespoke digital pathology algorithm to quantify sting localization and thus infer sting activity. This has shown that patients with high sting activity are associated with improved outcome in ER-positive breast cancer, and this is associated with an active immune infiltrate. Poor outcome perinuclear low cases are instead associated with an immune-suppressed M2-like infiltrated tumour microenvironment, which appears to be underpinned by amplification of MYC and also association with mutant P53. Therefore, we suggest that targeting either MYC or mutant P53 may be a novel treatment option in order to de-repress sting and lead to improved outcome. Finally, I just want to say thank you to everyone who contributed to this paper, especially Eileen Parks, Matt Humphreys, Elaine Gilmore, Stephen McQuaid and Manuel Saltateles. I want to thank our funders, Breast Cancer Now and CRUK, and also thank the Northern Ireland Biobank who provided the tissue. And finally, I also want to thank the rest of my group, past and present, at the School of Pharmacy at Queen's. Thank you for your attention.